Welcome, everyone. My name is Kelly Lamb, and I am the Director of Operations here at the MIT Enterprise Forum of Atlanta. We're glad you're joining us this evening for our first virtual event. So our agenda starts off today with a brief housekeeping tour for those of you who might not be familiar with Zoom, or maybe perhaps have never been to a webinar on Zoom. And after we've gone through this very brief tour, we'll kick off our introductions, followed by a panelist discussion with our experts today. And then prior to the end of the meeting, our attendees will have an opportunity to ask questions of our panel, which I will review in just a moment. So let's get right into it.
So thank you, Kelly. I'd like to thank everyone to this evening's program, COVID-19, a healthcare perspective, with our panel of experts on the latest in solutions for coronavirus. Also, thank you to our panelists and Gayatri for moderating. And for those of you not familiar, MIT Enterprise Forum of Atlanta is one of 18 chapters worldwide. Our mission is to inform, connect, and coach tech entrepreneurs, enabling them to rapidly transform ideas into world-changing companies. We're a global network of local organizations inspired by MIT and open to the world. Here in the Southeast, we focus on educational programs at the intersection of technology and business. Affiliation with MIT is not required to attend any of our events or volunteer with the organization. Before we get started, I want to take a moment to thank our sponsors. Without them, we would not be here. First, I want to thank our platinum sponsor, Evershed Sutherland. I'd also like to thank our annual gold sponsors, Miller & Martin, TriNet, Acuity, JP Morgan Chase, and Integra. As a follow-up to tonight's event, we have an event in the works for May. Tonight, we'll be discussing what we can do to save lives from a healthcare perspective. The event next month is to learn from economists about striking the delicate balance between saving the lives and saving the economy. And with that, I'll turn it over to Gayatri. She's Executive Director at Emory's Office of Corporate Relations.
Thank you so much, Catherine. Welcome to this panel on COVID-19, a healthcare perspective. We are all dealing with COVID-19, its impact on the economy, our health, and our life. Although economically it may seem very similar to what we just saw with the last recession, this is different. The 2008 recession was rooted in economics and finances. This time, the downturn is rooted in biology, a disease. Therefore, all the solutions that we put in place to address the economy and the finances will not be enough. We also need to come up with a solution that is rooted in biology. This panel is not going to talk about the clinical and healthcare delivery perspective. This panel is going to focus on the looming biological solutions, both near-term and long-term. Today's panelists are all working in some shape or form to get to biological solutions for COVID-19. Our first panelist is David Perryman. He is the Chief Operating Officer and Chief Intellectual Property Officer for DRIVE, the Drug Innovations and Ventures at Emory. DRIVE currently has an antiviral drug candidate in the clinic for coronavirus. Prior to DRIVE, David was the president, CEO, founder of Xyrus, a biotech company. In addition to Xyrus, David has founded 2 other biotech companies. One was acquired and the other became a publicly traded company. David has served on the board of directors for these and other biotech companies. And prior to joining Xyrus, he was a shareholder and head of the biotech patent practice group at Needle and Rosenberg, which is an IP boutique law firm. He practiced biotech patent law for 20 years, delivering patent business strategy to biotech companies, venture capital funds, the CDC, NIH, FDA, and numerous universities. My God, that's a lot. He also served as a founder and special advisor to Georgia Venture Partners, a state-associated biotech venture capital fund, And I believe if you look at the chat, David has pasted a poem he has written on coronavirus on the chat that you can take a look at later. If it's not visible, let me know and I'll ask him to paste it again. Our next panelist is Dr. Farshad Giraku. He is the Chief Scientific Officer at GeoVax. I hope I did not butcher his last name. In 2014, he was named one of the 50 most influential people in vaccines in Vaccine Nation's 2014 list. Prior to joining GeoVax, Farshad was CTO at Vaxis Technologies in Cambridge, Massachusetts, which was developing technology to help with thermostabilization of vaccines and biologics. Farshad has extensive experience in biotech and pharma world as a senior leader in vaccine research and development. He has worked at Hukipa Biotech, developing vaccines against cytomegalovirus and cancers. In Sanofi, he was the executive director of external R&D. At Acambus, he invented the platform to develop vaccine against several viral infections such as Japanese encephalitis, West Nile encephalitis for veterinary use, first dengue vaccine approved in Mexico and Brazil. Our third panelist is Dr. Rama Amara. He is the Charles Howard Candler Professor at Emory University. Rama is a leader at the Emory Vaccine Center and has played a major role in preclinical development of AIDS vaccine, now in phase 2 human clinical trials. Currently, Dr. Amara research focused on understanding pathogenesis and immunology of HIV/AIDS. His major focus has been on the development of novel prophylactic vaccines and therapeutic approaches to control HIV/AIDS. He's also working with collaborators in India to develop an HIV vaccine that could be used to control AIDS on the Indian subcontinent, where the strains of HIV are predominantly different from those in the US and other Western countries. Today, he will talk a bit about his work on coronavirus and the immunology around the disease. I would like to kick off the panel with my first question, and I would like David to kick off the answers around this, which is, why is coronavirus such a threat? Can you tell us a little bit about the virus?
Okay, I'm just going to repaste the poem because someone asked that. So I wrote that poem because, I remember back early in this, really watching it closely and being scared initially about it. And a lot of people were talking about, oh, it's just the flu. It's just like the flu. And I didn't think so. And it didn't seem like it from the news. And it didn't turn out to be that way. And so to me, the big challenge has been, how different this virus is and how unprepared that, in some respects we've been. Sort of like the start of this seminar. It's new, it's coming, it's something we're not used to, and we have to get good at it first. And it's been a stress test. So, that it infects easily, it spreads easily, it has a high mortality rate, relatively high mortality rate, spreads without you knowing. There's no immunity naturally to it because it hasn't existed. There's no vaccine to protect us. There's no antivirals to protect us. There's no drugs that have certainly been approved that can affect its damage that it causes on the liver. So there we are in a mad scramble. And so that's what, drive and we've been in and have been watching and trying to help affect.
Sure. Rama, what are your thoughts? I mean, you're coming from a very immunology, pathogenesis standpoint. Can you tell me a little bit about what you think is different about this virus?
Yes, first of all, I would like to thank Gayatri and the team for giving me this opportunity. And before we go forward, I also would like to say that my experience with this virus only for the past 2 months. I don't have experience working with coronaviruses before. And there is so much information coming out every day, and it is so hard to keep up with all of it. So I would try to talk about things that I have read myself and kind of try to scrutinize with my own eyes. So I'm pretty sure there may be some mistakes, so please excuse me for that. When I thought about this virus, first I know there are a lot of people, everybody is really worried uh, for the right reasons. We have about 2.7 million, or is it 1.7 million? It's 1.8 million, so 2.7 million infections worldwide right now. But when I think about this virus, personally, I'm not too scared. I think the reasons being that I feel that it is relatively easy to generate a vaccine against this virus. I'm coming from the perspective of what I have seen with its cousin, that SARS coronavirus that was an epidemic in 2002. I'm saying all this because I want first people to actually calm down. There's no need to be really worried. I know there are a lot of deaths unless you are at high risk. An older person or have underlying conditions like diabetes or hypertension. Don't worry too much. It's easy to tackle it as long as you follow the precautions. And my optimism comes from the hope that we may be able to generate a vaccine that is going to be effective, because the cousin of this virus is the SARS-1, I would say, is very highly immunogenic. And in the animal models, we were able to generate a good antibody response that could actually neutralize the virus. So I'm not too scared. I'm more optimistic, but one need to be careful. The thing that is very different about this virus for me is that people have a lot of virus without having any symptoms. That's what causes the major issue. So, for example, with the flu, somebody has mostly virus when they are symptomatic. With this one, so then they know that, okay, I'm not well, so I can isolate myself. Or people around that person would isolate themselves. But in this case, the study showed that people actually have the highest amount of virus 2 days prior to they developing symptoms. So that's what actually scares most about this virus to me.
Farshad, can you talk about what your comments are based on what you have seen so far in the coronavirus infections?
So yeah, I have worked with many different viruses, and I quite understand what Rama so peaceful is, or is more optimistic for a vaccine, because Rama has been working on a vaccine which you can call it the most difficult vaccine that you can ever work with, and that's HIV. So I know, because we have been working also on HIV in the last 15 years, and I agree with Rama that this virus, we can make a vaccine for that, but I think it's still important because we have about almost 200,000 people have died from this virus. And as Rama mentioned, the problem with this virus, in contrast to its cousin SARS, which came in 2003, and kind of company rushed in to make a vaccine. By the time they have some vaccine candidate, and they were ready, the virus died like 8 months later, and all the investment was disappeared. So, but this virus Rama mentioned is different because there are some people who are asymptomatic, they don't have any symptom, and you don't know who these people are. So you might have no issue with it because you may not have any underlying health condition and you are healthy and young and or maybe children and so on. But you can carry the virus and you go to your parents, you go to your grandparents, and you don't know that you are, carrying the virus because there is no large epidemiologic study done to see how many people are carrying the virus and what's the prevalence. So you bring the virus to your family, you bring to your father and grandfather. So you put them in danger. Yeah? I had a son in New York. He was studying and his school was finishing in spring break, and he was supposed to come to Atlanta and see me. And he said, "Daddy, I'm not coming to see you because I don't want to put you in danger." So it wasn't danger for him, but danger was somebody who is more than, elderly people. So this is a virus which has not been around. Everybody is naive to the virus. And the virus has this called ACE2 receptor, which is easily can infect human. There's no adaptation needed for the virus. The virus came directly maybe from the bats and maybe just animal reservoir, something like pangolin or so. But the genome is like over 99% is identical to the bat virus. So the virus didn't need any adaptation to be infectious and be transmitted in human. So it was easy to get into human, and everybody, billions of people are naive. So the virus has a long way to go until to reach, I don't know, 70, 80% of the population are infected. Then it can trigger the kind of herd immunity, then the virus can be transmitted in that way. Or before that, maybe we can find a vaccine which is effective, which I think we can make a vaccine based on experience with SARS and MERS. But we have to be very careful that the vaccine is safe in addition to be effective, because there had been some issue with the previous vaccine, which has not been safe. So we don't want to vaccinate healthy people, but put them in danger of, make the disease worse if they haven't been vaccinated.
So Farshad, just in line with the same point, so based on what you are seeing, do you think the vaccine will be more like the seasonal flu vaccine, or will it be meaning that it will be periodic doses over time, or will it be more like the polio vaccine where you take it once and you're done?
Well, I think the virus will not be like the flu virus, because the flu virus changes the antigenic. So every year the antigenic shift and drift causes a new flu virus. That's why you have to have a new virus made from scratch every year. So that's called the seasonal flu. So lots of people working on the universal flu, which you may not need to have vaccinated every year by going after conservative area of the virus to have a vaccine which can be used every year and not make from seasonal flu scratch. But this virus is different because it's not going on the antigenic drift or antigenic shift. So the virus is very stable. It's not like a flu. So if you make a vaccine for that, it will be effective in coming years. The question is that how long the immunity will last after vaccination. Is it lasting 6 months? Is it lasting 1 year, 2 years, or is it a lifelong? So nobody knows about that.
So Rama, I want to come to you with this question because people say that most of the mortality or most of the severity of this disease is because of your own immune response to the infection. So with that in mind, how are the different therapeutics and different vaccines going to come into play if your own immune system or hyperactivity of your immune system is going to be the reason why you get severity in the infections?
So we are still learning about what all things that happen after infection over time. So in any infection, there are phases, right? The early phase where the body fights through our innate immune system, where there is no prior memory or so these are the cells like dendritic cells or macrophages, and these are innate cells or NK cells. All these come into the play and try to control the virus by stimulating certain factors. And then if they fail to, and while this is happening, the body develops something called adaptive immune system. That's where people talk about the B cells that produce antibodies. And then the T cells, we have these helper cells and killer cells. So they generally come from day 7 onwards. So if the initial innate immune system fails to control the virus to some extent, if that's when the host loses control, and there may be too much immune response towards the virus, and that could lead to pathology. That's where it means that your whatever the response generated by your body will start attacking yourself. So it is when they say that, okay, there is an overactive immune response. So they're actually talking about at a later stage. They're not talking about an early stage. So it's hard to, right now, without understanding what all the virus does, it's hard to define the number of days into infection. So that's why they say that it is, you have to be very careful when you use hydroxychloroquine, for example, because if the individual is going into a state that there is an immune suppression, then you would use certain drugs. If they're going into a hyperactive immune response, you use a different kind of drugs. So the bottom line is that it is too early to be able to say how these things work. But in terms of the vaccine, how the vaccines work is they act very early, even before the virus take off. So that vaccines certainly ideally should not give an opportunity for an overactive immune response towards inflammation.
Thank you, Rama. I'd like to turn the topic towards the therapeutic options that are currently in the works. And I think, David, if you can talk about not only the drive drug that is the clinic, but any therapeutic solution that you think that are in that look promising.
Okay. Well, to the optimistic point, for a biological solution or a chemical solution as part of biology, there are 226 drugs according to BioWorld in development currently. Right, that's a lot of firepower. That's a lot of people coming after this. So that's very encouraging, right? Our industry is so much more advanced than back in the day, certainly back in the day when I started. And so there's a lot of room for optimism. But those to the point Rama made, it's sort of the type of drug depends on what you're really doing. So there's this class of drug called direct-acting antivirals, right? And so they are in some way directly interfering with the virus's proteins, the virus RNA, its ability to replicate itself. And there's drugs that are based on host factors, right? The virus infects us. It hijacks our cells. It hijacks our cellular machinery to reproduce itself. And there's all points in there where we could intervene with the host protein, our human proteins, so that the virus can't replicate. And so there's examples of those there. And hydroxychloroquine may be in that class, but in terms of blocking viral entry, but it may also be an immune modulator. And it would kind of, when you use it, you would use an antiviral part of it early in infection, and then probably later infection for immune modulation or suppression of the cytokine storm that comes with this. And so that's really tricky. That drug has had, a lot of press around it. It's interesting. It's a repositioned drug, a repurposed drug. And the company I used to run, Xyrus, was around that concept. What are the host proteins that are really essential for broad spectrum viral replication? And then how do you, sort of tap that knowledge to create new drugs? And so we'll see on that. You know, it's mixed. The NIH Tony Fauci, et cetera, just came out with a report today where they're basically not endorsing or denying any antiviral drugs. So that's in that class. Then, you know, the ones that we naturally like at Drive and that really have been the cornerstone of antiviral therapy over the years are the direct-acting antivirals. And in our case, they're drugs that directly interact with the viral polymerase, so that when it's going to reproduce itself, its RNA, it induces mutations. It inserts the drug rather than into its nucleic acid. That causes catastrophic mutation, and the virus ultimately goes away. And those, the real benefit of those, and remdesivir, that's also in clinical testing, is in that class, and a lot of potentially good press coming out of that. The beauty of those is that they're potentially very broad spectrum. So broad spectrum can be within coronaviruses, or it can be beyond coronaviruses. But this polymerase that the drug's interacting with is very highly conserved in RNA viruses, which coronavirus is. And so one drug could come in and potentially work against a multitude of viruses. So the beauty of that, both in terms of proactivity for coronavirus and in general, is that you can make potentially, to the proactivity realm, a couple of drugs, 4 or 5, you name it, that are broad spectrum that cover most RNA viruses. And they can be there, they can sit on the shelf, and they can be ready for whatever emerges. So that's interesting. And our story, and that's promising, because that's something that's, you know, with some investment you could have. And we have one that's in that class potentially. Ours works against Ebola, chikungunya, influenza, coronavirus, so, and others, and biodefense applications. So those are very important potential drugs. And we'll see. So remdesivir, the thing about these direct-acting antivirals is you want to go in early in infection. That's pretty much been the cornerstone of antiviral drugs. Remdesivir is being used a lot late in infection. And the reason is it's IV. So you have to think about not only the type of drug, but how it's administered. So if we're in a pandemic and we all need to you know, quickly get this drug on board if we have infection, so that we stop spreading it, so that we don't go to COVID, so that we don't get extremely sick. Yeah, you know, IV is not really the answer. Hopefully we're pulling for it. It saves lives. It's a great class of drugs. Ours is oral. So it's early. We just entered human testing. But it's come out of 4 years of work, $35 million of proactive government spending to help us develop the drug. It's been in a lot of, you know, the top journals. And so we're very hopeful that we can, kind of like in the horse race of these 626 drugs, some drop off, some keep going, and we hope that ours can kind of come from behind and help a lot of people.
Sure. So that's the therapeutic standpoint. Farshad, I know GeoVax is working on its own vaccine for coronavirus. And can you talk a little bit about that, as well as the Moderna RNA virus that's currently being tested in the clinic, or any other promising vaccine that you have heard of?
So I think like any vaccine or any virus, you know, that you want to target, there are a series of tools that you can use to make a vaccine. One of them is using the DNA approach, just injecting DNA of the corresponding virus by an electroporation or a device, or just single syringe and needle. Or the new approaches is also nucleic-based vaccine, is RNA. Everybody knows about the messenger RNA. which has been around for a long time. It has not been stable, but recently it has been, formulated in nanoparticle and has been stable. So it can be used directly to inoculate into body, and that messenger RNA code for a specific protein, which is known to protect against the related vaccine virus. But there are also protein-based vaccines. You take an antigen of the known virus, which you think it would be protective, and produce it in CHO cells or baculovirus and produce a vaccine made from the protein subunit. But there is also vector-based vaccine approaches that we are also using, which you use another virus to deliver part of the vaccine antigen, or the vaccine, in this case, coronavirus. So what we use, our approach is we use a very safe vaccine virus, which has already been tested in the last 50 years. And we use that virus to deliver a pieces of this coronavirus as an antigen. So induce the immunity to this piece. So when the real virus comes to infect people, that virus provide protection, because the body has already seen the pieces of the virus and recognized the troops are already for action, and they induce antibody to block the entry of the virus, but also they induce T-cell response. So if the virus get into infected cells, they quickly eliminate the infected cells and the virus reservoir. So this has been our approach. We think we can deliver not only one antigen of the virus that everybody else is using, the envelope protein or this S protein, spikes protein, which can give the name of the corona because it looks like a crown. This is the major antigenic target, but we are also using 2 other antigen from the virus. And it makes it about a large piece of the genome we put into our virus. And it's about 6,000 or more nucleotide. And coronaviruses are one of the largest RNA viruses. And they have a genome about 30,000 nucleotide. It's huge. So we use about 6,000 to 7,000 nucleotide and put into another virus, and that virus is capable of delivering this large amount of genetic material, which is needed for protection against the coronavirus. And we hope the protection will be broad, because we not only have the spikes protein, but we also have other components which can bring the epitope, which is broadly conserved among other coronaviruses. So if next year a similar coronavirus comes, which you don't have the vaccine for, maybe we can provide some protection, not 100%, but some protection.
Sure. I mean, now we are also doing this antibody testing to figure out who in the community have been infected. And just today, I saw an article come out saying that the Roche CEO is saying COVID-19 antibody testing is a disaster. I mean, can you talk a bit about why somebody like the Roche's CEO would be saying that it is a disaster? I mean, the testing, is it because we are not using all the regulatory strictness before we approve those tests, or? Well,
I don't know about the Roche test, but I assume if you have a rapid antibody test, like the one you do for pregnancy or HIV, I assume you get some saliva or maybe finger prick in blood from finger, and you get the answer, you have antibody, yes or no. Mm-hmm. Okay, so it doesn't tell you anything if you're protected or not. The other way to do that is, measuring the antibody, like they call it ELISA, the binding antibody to corona spike protein. That gives you kind of quantitative measurement, how much antibody you have. But, and the best thing would be, we call it neutralizing antibody, which measure what is the level of the neutralizing antibody in your body that can neutralize the virus. So that gives you maybe a better correlation or guess if you would be protected or not. Again, the protection, or we call it correlative protection, is not known at this time. Is it the antibody only? Is it the cellular response you need for protection, or you need both of them? It is unknown. But some people think, the antibody could give you some protection. That's why, the immune globulin approach, they take, serum and antibody from those people who have been already recovered, and they purify that, inactivate it for other viruses, and try to give that approach to try to help, just like therapeutic. But this immune globulin antibody approach has been around for at least 50 or 60 years. But the problem is that you have to have a placebo-controlled study to show if that antibody approach is working. So just giving the antibody to 100 subjects and see, okay, maybe some of them recovered, some of them not, is not going to give you the answer. You have to have the placebo control. Let's say giving to 500 subjects the antibody, and the other 500 subjects are getting nothing. And then you can see, what is the reduced number of infection, and then you can. This is the same thing happening for all these drugs. They say, okay, chloroquine is great, or IL-6 is great. I'm giving to these people, and some of them survived. It's great. I'm really excited. But as long as you don't have the placebo control, you cannot get the meaningful answer if this thing is working or it's just anecdotal.
Right, right. I'm going to start looking at the questions that the audience has put in. So I know you already touched on it. One of the questions is, can you develop immunity if you get it once? And this is prime in everybody's mind, right? Okay, if you got it, even if you were asymptomatic, does that mean you're protected? And based on what you just said, we don't know enough to be able to say we are protected. Can you, is that right?
Yeah, I think you don't, as I said, you don't know the correlation of the immune response. You don't know the correlation of protection. You don't know if it's only antibody or it's a cellular response. And if the antibody, if it's neutralizing antibody or the other source of antibody which have some functional activity. And until that is clear, you cannot measure that. Yeah, but I expect that those people who are infected, at least they will be protected for some period of time. The time is not clear. Is it 3 months? If it's 6 months, if it's 1 year? You know that with the similar coronaviruses, which is causing common cold, you can get common cold again and again. So you are getting some responses, but next year you can also get infected. But assuming if you already have infection, even if you're infected, your symptom will be less and your duration of the illness will be shorter.
Sure. And also that, I know, even for the assays to measure neutralization, there can be multiple different versions of them. Mm-hmm. Then I think people are still, everybody's developing their own assay. Some use the actual virus, and some use a pseudovirus, only a part of the virus for the assay. And the results are going to be very different depending on what you use. Yeah. It's just like the issues that they have with with the serology assays. They will run into problems in what kind of neutralization assays that would actually correlate with protection. So there's going to be a lot of information that need to be built to be able to really arrive at some correlate of protection.
Exactly. So what is that WHO is doing, is running a huge clinical trial called Solidarity with lots of vaccine company. And what they need to do have to standardize the assays. So have the similar reagent provided to the manufacturer. So everybody using the same antigen, the same standard protocol, and measuring the same thing. So then you can compare different platform and different level of neutralizing, as Rama said, doing with pseudotype or real virus or subunit kind of thing. It's giving you different level.
Right. And I think there is another question now with the latest thing coming up saying that 55 times is undetected. So David, I think somebody has asked this question to you specifically. Do we really know what the fatality rate is, knowing that 55 times is undetected? Can we really say it is more has higher mortality than influenza, or can we just say it is more infectious? And so,
Yeah, it's a good question, and it's been sort of my pet question from the very start of this, because originally, I mean, there's a lot of other factors, right, that aren't mortality, right, which is that we don't have any immunity, that there's no vaccine, that there's no drugs, and that it also causes lung damage that flu doesn't cause, et cetera, et cetera. But specifically to the question, originally my thought was, and I thought the Diamond Princess was the best sort of controlled experiment, because you knew exactly who was infected and exactly how many people died. And that came out about, a little more than 1%. I haven't followed it lately. More people might have died because they were still critically ill when I was looking at it. And so that gives you some idea in a relative test tube. Now those patients were older, there's multiplicity of infection issues. So we don't know exactly where it'll end up. But I think the fact is, even if it's, and you could also question that data on flu, right? I haven't gone back and looked, but this 0.1% lethality in seasonal flu, well, are we detecting everybody that actually has flu, or do a lot of people attribute it to the common cold that never gets reported? I don't know how statisticians deal with that ultimately, and we'll have to deal with it here, but the numbers in the future will definitely bear that out. But I think it's interesting, but I don't think it's the main thing of why coronavirus is such a threat to us. It's part of the threat to us. And flu
So
is
when
bad.
you do, so when you do just did simple calculation, if you look at the Johns Hopkins website, you take the number of death and divide it to the number of infection which is reported. Now you get about mortality of 6.9%. So this is high. So but the number of infection could be 10 times higher, or maybe 50 times higher, because we haven't tested everybody. So we don't know. So if that's the case, the mortality or CFR case fatality rate become much lower, maybe become 0.6, and that comes in the range of what flu is. So in that range. So now we have 6.9, and apparently this is the overall. It is higher the older people and lower in younger people, but overall it's about 6.9% if you just calculate the number as it is now. But once you know the number of the infection, the mortality rate probably coming lower.
Right, but yeah, and I'll just sort of add in those, I mean, people have run models, projecting, trying to factor all those, and 0.6 to 2% is where I've heard people coming out. But even then, 0.6 is 6 times flu, and seasonal flu, at least swine flu, maybe 0.2, so 3 times. Combine that with the other factors, and that's why we've got the problem we have.
And that makes sense for viruses to survive. The viruses, they don't, if they want to survive, they have to not to kill their host so quickly. Something like Ebola, you kill about 60% of your subject. So it's easy to control the virus, because you isolate the person is dead, and the virus is dead with the person. In order to survive, you have to come to a low mortality, which you can spread. And if you have some subject which is asymptomatic and you don't know who these subjects are, this is even worse. They can spread the virus, and the virus stick around with us for a long time.
Right, but I would just sort of add, as you know, it also depends on how long it takes from infection to symptoms. If you look at HIV, highly lethal, but it didn't eliminate people because it incubated so long before it caused the damage. So this is lasting longer than flu. The symptoms on average, that adds to the problem. What you raised earlier, asymptomatic, also adds to it.
there is one question that talks about why there is not more examination of introduction of various metal ions such as zinc, copper, or gold, which have seemed to have shown some successes through metal ionophores. And hydrochloroquine, for example, has been examined in trials in its own merit, not as a zinc ionophore. So any of you, if you have any thoughts on this?
Well, I used to take zinc at first signs of a cold, so I studied it a little bit, and so I'm not expert in it. But there is some clinical studies that I saw back then, and there's some logic behind it. Then when I talk to a rhinovirus expert, they say they absolutely dismiss it. And so all these things that are not, to the earlier points of Fashad, controlled clinical trials done properly, double-blind, placebo, et cetera.
Yeah.
You just don't know. You're not going to know, and it might work, or it might be, a form, at least in this case, of snake oil and false hope, and people get sicker, like that retrospective study on hydroxychloroquine that just was published today. It's retrospective, so how much do we put into it? But that was saying that the death rate was higher with hydroxychloroquine than with nothing.
Yeah.
So it's risky.
Yeah. So
Yeah,
what do you got to lose? Yeah.
Again, it is very important to keep in mind when you start using the drug, also makes a huge difference in what the outcome is going to be. And a lot of these drugs, if they're used very early in the infection, I think they would be effective. But once you start using them late in the infection, they could backfire.
And I've also heard about, sorry, I've also heard about the infection being two different kinds. One is upper respiratory and then a lower respiratory, which is more severe. So I see a question that came up from the audience that, can you walk us through the stages of the infection for COVID-19? Like what happens? A lot of people are asymptomatic. Are they only getting sinus infection? Whereas the rest of them who are more seriously impacted, are they getting a lower lung infection? Or is that not true?
I don't know whether there is enough data out there to really see where the infection is localized at different stages. But what I think is striking is it is mostly seems to be the upper respiratory infection, and then it actually can persist there for a long time. And the studies have shown that people can actually become negative in the nasal swab, but the virus can still persist in the sputum and in the stool for up to 35 to 38 days. So that's actually brings up this question about when people say that, oh, they became negative and then the same person became positive again. It is important to check when they say they're negative, what part of the secretions that they were actually measuring. For example, if they measured in the nasal swabs, nasal secretions, and they can be negative, but we do know that the virus can still be there in the sputum. So these studies need to be done carefully, and then interpretation should be thorough before one can generalize them.
Right, and I'll just add that not being a doctor, it looks like it's infecting much more broadly than just in the respiratory tract, right? There's neural issues of loss of sense of smell. There's, hepatic and renal dysfunction. And so those are not things I don't think we see with flu. And so that's to me, that's an indication it's much more infectious broadly than the respiratory.
Yeah, that's a good point. And also the receptor, the ACE2, is also expressed at very high levels in the intestine. So that's why you do see a lot of virus coming through the stool. So we may need to have vaccines that actually also protect you from the infection in the gut.
That's interesting. Today, I was listening to Chumakov, Dr. Chumakov. Some of you probably heard the name of Chumakov. He was his father developed the polio vaccine. So now he's working at FDA as a scientist. His son, his name is Kostya, and he apparently working with NIH and others. They want to use the polio virus as a kind of countermeasure against the COVID-19. So the idea is that all live viruses, they can stimulate the innate immune response that we spoke today. And they can give you kind of non-specific protection against other viruses. So the idea is that give them, the oral polio virus, and they will be protected for a certain period of time against the coronavirus. So the polio virus, oral polio virus, has been approved, but the only problem with oral polio virus is that the reversion and the adverse event that you can kill the subject in, I don't know, the rate is maybe 1 in 100,000 or so. But again, you have to weigh the cost, the benefit, and the problem with it. And you can use the polio virus or yellow fever vaccine, or you can use the BCG vaccine to induce innate immune response, so then can protect against other viruses. But this is really a short measure. This is not the true specific vaccine.
There is one question that talks about the antibody tests that are in the works. Is there can anybody comment on the sensitivity of the antibody virus? But I think you already commented on, there are two ways to look at it. One is to see whether we are creating the antibody. The other is to look at whether they are protecting you. But in terms of either case, you need to make sure that they are sensitive, right? So do you have an idea of how sensitive these tests are? Like what titer do you need to have to be able to measure it?
No, I don't know. I think there are so many antibody tests out there, and FDA has kind of loosened, the requirement for approval, and some of them are false positive and some of them are false negative, and the different sensitivity. And it depends, what antigen that they're using. If it's, the whole S protein, or it's a fragment of the S protein, what is the cross-reactivity with other coronaviruses? What is the specificity? I don't know about all of them, but they're all different.
And then I generally think if a test that is designed to get a rapid result, very likely to have less specificity or sensitivity, and this combined with immunology of these antibodies is in general is these primary response. Early on, these antibodies have less affinity, means how tightly they can bind to the antigen. So and then these tests need to really keep that in mind, so that, and when you have low affinity, you generally require a higher amount of antibody to be able to score positivity. So all these factors come into the play. I think that's what is causing these differences between different tests.
I know we are running out of time, but there were some good points about, there is another drug that is similar to remdesivir, which is Gardasil. And somebody said, has anybody thought about that? Because that is an oral drug. The next one, somebody said about, going about it as a combination therapy, like you go with remdesivir and then also give an anti-inflammatory or look at it from a staged approach. First, you give the remdesivir, and as the disease progresses, you can give anti-inflammatory as needed. And then lastly, there is a question that somebody said, when will the panelists stop sheltering in place? That is like really asking us, when will we stop doing it, so that, that gives them an idea of how.
Well, I still go to work every now and then, but we try to work in shift, so we don't come in contact with each other as much as we can, maybe 1 or 2 people. And I still go to do my shopping. Of course, I use mask when I go there, and you clean up, the cart that you're using it, And I try to use mask and maybe gloves as much as I can.
And I go to work every day, primarily because that I don't work at the bench. I'm mostly confined to my office. There are a few people in the lab that are actively working, try to push things forward, and they actually wear masks. And then when I go and talk to them, I always wear a mask. So you follow these guidelines to make sure that we don't pass on anything, or we don't pick up anything.
Yeah, the virus is supposed to be in droplet, which, they say that's 6 feet, but there's some study which shown, there's some transmission by aerosol, which you might need more than 6 feet, maybe, 15 or 20 feet. So it's small aerosol, which can carry the virus to a larger distance than 6 feet. So you have to be really careful.
What about you, David?
Well, I'm lucky enough to be able to do most of my work from wherever I am. So, it's a practical thing. It's a risk benefit. A lot more data is coming in. I'd love if we had a more advanced antiviral, some of this data to come out. If we had great testing, I'd feel a lot better. I see people, just habitually getting too close, even when they're on the street, and that's a little scary to me. And things that are going on now, to me, is too early. Some of the governor's stuff. And so, I'm just going to have to, risk benefit and wait and see, and hope the longer we can go, the better off we'll be, because testing will be better. The drugs will be more advanced. We'll know more about them. And hopefully these vaccine guys will come in the long run and save us all.
Yeah. And before, it's already way past our 6:30, which was the allotted time. One last question that did come in is the seasonality of the coronavirus. And I just saw today the CDC director saying that this time, if we think this time is bad, wait for the next season when it'll hit along with the flu. So yeah, I mean, yes, we have to be optimistic, but we need to be sensible. And like Rama says, and like Farshad and David, as long as we are wearing our masks and gloves and taking precautions, and don't go in large gatherings, just be watching.
And get your vaccines. Get your vaccine. They may help.
Yes.
We talked about the innate immune response. All the vaccine are stimulating the innate immune response, and you protect you not only specific against that virus, but the viruses don't like to be together, so they don't like to coexist. So one can just eliminate super infection by a second one.
Right, and I'll just add too quickly to the point about masks, because I made a post a long time ago about masks, and culturally or for whatever reasons that wasn't going on here. And so I just sort of, I think we just all need to use our heads and our common sense, right? It makes sense that masks would prevent both ways. And there was actually some data on that. There was a pretty good paper on that. And for the longest time, it was resisted. And so, do the things that you think are smart. Don't worry about feeling awkward in a crowd, or, whatever our human natures are. And stay safe, everyone.
Yeah.
Thank you, panelists, and thank you, the audience. Catherine, I'm going to hand it off to you.
All right, thank you so much, Gayatri. And again, special thanks to our panelists. Now I know I'm going to wear a mask for a really long time, and our sponsors for making this event possible tonight. So don't forget about our May event, where we're going to be discussing the virus's impact from an economic perspective. Also, be sure to head to our website to subscribe to our newsletter to get an update on all the future happenings at the forum. We're also on LinkedIn and Twitter. So thank you again, and we look forward to seeing you at the next event. Thanks, guys.